All right, welcome back to video 2. So you've just started looking at the process of DNA replication. You've done your daily drill. Let's dive into what's actually happening in this process. All right, so as always, you'll need your notebook, pencil or pen, and your curiosity. Our guiding question. So how does the cell regulate the process of DNA replication to ensure the daughter cells have identical genetic information as the parent cell? And thinking back to the cell cycle in general, the goal of the cell cycle was to make an identical daughter cell. And it's not just to make an identical daughter cell to make an identical daughter cell. The purpose was to replace a dead cell or a damaged cell so you can reinsert it so it can complete the functions the previous cell was doing. For example, let's say I cut my hand. When I cut my hand, I lose cells. My cells are dying or dead or damaged. And so my body is going to use the cell cycle to replace those cells with new cells. I want those cells to be identical. I don't need a nose growing out of my hand. I want it just to be skin tissue. So again, we're making identical cells to replace the damaged cells, damaged or dead cells. And so DNA replication is a part of that. And so in order for my cells to grow back and look just like the skin cells on my hand, DNA needs to be identical. And so that's where DNA replication comes into play. We've learned about Chargaff's rule. We've learned about we learned about Watson and Crick and how they discovered the structure, this double helix structure. And so now we're going to jump into, so we have this DNA. It's wonderful. It's in our cells. It's a huge amount of it. How do we copy it? And so in your notebooks, pause me, whatever you need to do, but I need you to make sure that you write down the goal. You should always know the goal of a process. Why should we care? Yes, DNA replication is something you need to know for this class, but why in general in your life should you care about DNA replication? And it's because we want to make sure that all cells have the same exact DNA as the one it's taking its place for. If we didn't, we would look like a crazy Picasso blob of things coming out of all the places that it shouldn't be, because the DNA would be, well, random. And so, like I said earlier, so in turn, the entire cell can replicate during the cell cycle to replace that dying or damaged cell. Make sure you have this in your notes. And so when we talk about DNA replication in particular, DNA replication, as a reminder, happens during S phase, because that's when our DNA is replicating and we're looking at chromosomes. These are the big players involved. We have our DNA strand, our double helix, just like Watson Crick figured out with help of Franklin and Chargaff and Pauling. And we have these three enzymes, helicase, polymerase, and another polymerase. But the big ones, just make sure you know helicase and DNA polymerase. This entire reaction is an enzyme-driven process. We should know that these are enzymes because they all end in, oh yeah, that's right, I can't hear you, ACE. They all end in ACE, and because they all end in ACE, we know that they are enzymes. So they are proteins that drive the process of DNA replication. And so let's look at what this process actually looks like. This diagram you absolutely need to have in your notebook. So I want to make sure you pause me at some point now, after, to write this down. We start with our original strand of DNA. This is our original or parent DNA. Our parent DNA in this case scenario is going to be green. Original DNA needs to be copied. In order to copy it, our helicase, so our first enzyme, is essentially going to unzip it. You can think of it like a zipper. I have one right here. It's stuck together. The helicase is going to unzip it. And so the unzipping means that it's going from this twisted ladder shape to two strands of DNA. One on the top and this one on the bottom. And so when the helicase unzips or unwinds the DNA, we're able to add new bases to it. And so this red enzyme, which is called DNA polymerase, is going to add those nucleotides to this two strands of DNA. So our two original strands, for example, have G, 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 T, is going to get new nucleotides, and the DNA polymerase is going to add a C that matches with a G, another C that matches with a G, so on and so forth. And so this new strand will attach itself to the parent strand or the original strand. It will happen on both sides simultaneously, so at the same time. And eventually what you're going to end up with was 2 identical strands. However, these 2 identical strands have one strand that was the original parent strand and one strand that is the new strand. So we call this semi-conservative replication. So semi-conservative replication is when you have half the parent strand or the original strand, which I wrote down here as this green, and half of the new DNA, which is the pink. It's kind of pink. It's supposed to be pink strand. And so that's essentially what you need to know for DNA replication. You need to know there are 2 enzymes, helicase unzips, and DNA polymerase adds the nucleotides. You're making 2 identical strands of DNA from that parent strand. And just a couple terms you should know: semi-conservative replication. The newly made strand of DNA is half the parent strand and half the new strand. And the anti-parallel, like we talked about yesterday, is when those strands are running in opposite directions. So I'm going to go back. This strand is being made from the right to left, and the bottom strand is left to right. You don't need to know that. You just need to know they're running in opposite directions. Sweet dance move, right? And so those are the big things you need to know. Make sure you know those enzymes. Sorry, I thought I saw something. Now you're going to go back to that worksheet and you're going to complete part 3. You're going to use your notes to apply your understanding questions about DNA replication, and you're going to make sure you press submit in that upper right-hand corner. When you're done with that, you've got your exit ticket. I'm on Zoom. Please jump into that phone call. I've had so many people being like, oh, that makes so much more sense now that you've explained it. So jump in onto that Zoom phone call. Ask a question, just say hi. Love to see you guys. And stay awesome, stay safe, and I will see you tomorrow.